I'm going to talk about a vaccine adjuvant from University of Tokyo. And I work on mostly, although I'm a professor in university, I recently work on with the children more for this SDGs, because kids know all this 1 to 17 of SDG, but we don't know. And 100 Days Mission is also another missions raised by politicians and policymakers. So we are between with the hardest questions and missions. And then don't Google myself. My name is Ken Ishii, and I'm the very famous DJ of heavy techno. So if you want to listen, you can try. So please put the J between so that that becomes my name in a scientific environment. So please remember that you don't find me in the Google. And why I'm from Japan, working on a vaccine R&D. So due to this 100 Days mission raised by UK government in 2021 with CEPI, and Japan realized that they didn't spend any money on this. And then they spent huge money on this vaccine R&D towards the 100 Days missions. And then they made a big SCADA center in the biggest funding agency called AMET. And Japan initiative world leading vaccine R&D centers are made. And we got selected as a flagship center led by Yoshihiro Kawaoka as a utopia. And we also support from Vaccine Design Center to this. And with 4 different synergy centers with the very famous vaccinologists and virologists in the university. And of course, Dr. Sho Yamasaki and Dr. Ueno is involved in this synergy center and support institutions. Utopia Center is led by Yoshihiro Kawaoka and with me and then Yasushi Kawaguchi and others, and then 41 PIs in the University of Tokyo. It was a couple years after I moved to University of Tokyo, and University of Tokyo had many famous scientists from AI and wearable sensors and all this, quantum science and so on. So these people actually volunteered to be a member of this center. So we don't know what's going to happen to, but as Dr. Gao said, basic researchers are now joining to do something for the vaccine and infectious disease. And this multiple research center is supported by many collaborators from all over the world, and we are now working on it. But you may not know what went on the vaccine R&D in Japan, because we only knew Pfizer and Moderna and other Chadox and so on. But Japan has actually started plasmid DNA, attenuate virus, recombinant protein adjuvant, LNP messenger RNA, and self-amplifying RNA, and plant-based VLP. Those are all led by all Japanese companies. And some of them are already approved. And then self-amplifying RNA was the first approval obtained in the world, and followed by many farmers. And also AstraZeneca and Novavax work with the Japanese company for production of. And I was deeply involved in this LNP messenger RNA approved. So since US is blocking this mRNA vaccine for the Moderna and Pfizer, Japan has been free of making any vaccines using LNP messenger RNA. So if you are interested in, we can collaborate with you. But I don't work on mRNA anymore because it's already standardized, industrialized. Now I'm working on EV vaccines, which is the plasmid DNA based, targeting this EV membrane proteins together with the protein of your interest of antigen fused, so that you can lead antigen to the EV, so that you can use this RNA or DNA vaccine targeting EV, but also you can collect these cells secreting EV and then collect this EV to the vaccines. So we are trying to make this as an intranasal or mucosal vaccine for this matter. I don't have time to talk about this detail, but with Utopia, I'm working on this EV vaccine. EV or virus or many small LNP messenger RNA, 100 nanometer particles, So we realized that the old style flow cytometric analysis can make analysis of single nanoparticles. I don't go into detail, but the flow cytometric analysis of nanoparticle has allowed us to use to conduct single nanoparticle biology. And we don't go into the PBMC or serum anymore. I mean, we do, but in addition to it, we now starting collecting your breath. And in the many different cities in Japan, and also France and other countries, with this interesting material called this R tube. And then air breath is full of the nanoparticles, but not so much cells, unless you are lung cancer or lung disease. But you can collect so much information from the air breath, and targeting not just the protein by antibody, but small dyes targeting lipid and nucleic acids and sugar chains, you can actually make a mapping of this nanoparticle from your breath and try to understand the immune response to it. So basically what I want to say is, in order to do innovation for the vaccine science, I don't work on a vaccine or immunology or virology, rather work with some other people in the cutting-edge scientists for the achieving 100 days missions. But there's another program called New Generation Vaccine, including new modalities. And so in our institute, with Dr. Keisato, assistant virologist, or Dr. Sorimachi or Dr. Tsumoto, who are actually protein engineerists and then also cell biologists, or mucosa immunologists, or people from Bastu, working together for vaccine design and so on. What we do is actually many researchers, non-infectious disease researchers complained that you guys had many BSL-3, but they never let us to use this. And in order to prevent this, we now work together, releasing all this BSL-3 facility to the many different researchers, but training them, starting from the surveillance of pathogen, vaccine design, and small and large animal experiment with these CROs, so that rather than publishing paper, we develop and make a data for the regulatory agency immediately. And that prevents us to understand combining adjuvant or delivery system. So we collected all these different scientists from all over Japan, especially for this adjuvant and DDS doing research, including Dr. Shoya Masaki is here. And then what we did, actually, it took 10 years starting from before COVID-19, about adjuvant database. Adjuvant database is actually now published last month. So you can try to look for what's going on in this database and publish paper. But basically what we did is we took the 25 different adjuvants, injecting them to mouse and rats, and then old-style transcriptome, because we had a huge toxicogenomics data done this with old style. Have you worked with a toxicologist? I'm an immunologist, and I love black 6 mouse. But toxicologists hate the mouse and loves this white rats. So it took 2 years for us, which one animal model is good for the adjuvant database. And then we never get along. So we chose to choose 2 animal models to inject this, all these different adjuvant into, and then collect these old organs. But turned out to be very good to have 2 different animal data. And I don't go into the detail of the adjuvant, but most of them, like aluminum salt to the CFA to the different ASL series and so on. So you can go through the papers. But what we learned is actually we took the lymph node, spleen, and liver, and then collected. So by any means, by transcriptome, you can just predict what kind of adjuvant you inject. However, the key was the series of genes. So draining lymph node is full of immune response. Spleens actually a lot of RNA metabolism. Liver, in a sense, has no genes regulated for the immune response, but rather whole metabolic genes were regulated. And we took these metabolic genes to understand and predict what is going on in adjuvant. Because you know, if you have experience for development of real drug and vaccine, liver toxicity is a real key bottleneck. And we had a huge national database, of open toxicogenomics database with 150 compounds, which is approved drug. And we could combine this adjuvant database and compounds data. And then, in addition to gene expression profile, hematology, biochemical tests, and serial microRNA profile were all dumped into the AI or old-fashioned machine learning. And what happened is that comparing toxic group or non-toxic group. So toxic group is similar patterns to, for open toxicogenomics of approved drug into toxic group and non-toxic group, and then compare with this adjuvant database. And interestingly, 1 out of 25 adjuvant became crossed by toxicologists due to this liver enzyme, which was a not ligand. It says adjuvant X, but you can say that this is, I will disclose this later. And interestingly, this adjuvant was inducing huge necrosis in the liver. And all the pathology says you cannot develop this drug as adjuvant. And then this was very interesting that it is correlate with some of the genes with this toxicology and then liver genes, and then this knockout has disappeared. If you make a knockout on mice of these genes, this necrosis was gone. And I'm sure that you are not interested in toxicology, but so you want to know among these 150 approved drug, can it be, or can any of these 150 known compounds to be adjuvant or not? And we predict this by combining this data, and then collected this by training by machine learning, choosing some of this interesting gene from the liver. And most of them are cell death and metabolism genes. And these are the gene sets. We took it, and we trained a set of the database. And we realized that some of these interesting compounds has a very high score that shows it's going to be adjuvant. And then we looked at, comparing to this ova plus alum, there's some equivalent to the alum inducing the total IgG to the co-injected ovalbumin. What was it? That was colchicine, one of the oldest drugs for the gout. And we are so excited. Is this colchicine can be an adjuvant? And we prepare our patents, and we made a team to develop colchicine as adjuvant until young student found the paper in 1991. Colchicine is a potent adjuvant for eliciting T-cell response. Thanks. So there was nothing new. that we just simply didn't know that colchicine was adjuvant. So this was the story of this adjuvant database, and tiny part out of this adjuvant database. So if you want to play with this huge database with toxicogenomics, you are free to use this. So we have a database for adjuvant from a different field, including NIH one. But this is very interesting because it's combined with open toxicogenomics database with 150 compounds. So, and multiple organs with the 2 different animals. And in this case, transcriptome pattern successfully distinguished each adjuvant regardless of organ or species. And a machine learning model can predict adjuvanticity and hepatotoxicity. And as a result, we found colchicine is the adjuvant, and FK565, developed by, which is a not ligand, was beautiful adjuvant, but turned out to be negative due to the toxicity in the liver. Overall, ADP combines with the OTG offers a framework for transcriptome-based data-driven screening adjuvant candidate. For 100 days missions, I keep asking ChatGPT. Yesterday with ChatGPT 5, I didn't put in the fridge. But the AI said, in order to make vaccine available to you safely and effectively, is to have, you have to have a pre-pandemic research and development, surveillance and early detections, regulatory framework, and a manufacturing readiness, clinical trials, data sharing. So never reached to the level that Dr. George Gao. But ChatGPT-4 was clever enough than 3.5. In order to make vaccine in 100 days, ChatGPT, okay, not me or Gao, Dr. Gao, said you have to have a collaborations. And more importantly, public engagement communication for the sake of transparency and educations. And this tells us that we as a scientist or a medical doctor never care about children or public outreach before COVID-19. So that turns out to us that next time in a pandemic, we have to work together to share all the information with huge transparency and a big effort to the educations. And I'm sure you don't have you have a full schedule for next month, but if you're interested in just traveling Kyoto or escape to the Kyoto next week, which might be very hot, you can join this vaccine meeting. And we have also a subsequent infection immunity meeting. But of course, you don't have any time, I know. So if you are not, if you want to plan to come back to Japan in a year, we have a third nucleic acid immunity meeting in Nara, September 16, 18. So please write on the text. And I'd like to thank to my team, which now expands to 25 people, but with the same missions, 100 days missions, and towards the kids, 1 to 17 of SDG goals. And Utopia is full of this old men like me, but diversity and inclusion is definitely more important than ever to us. Thank you very much for your attention.
Thanks, Ken. Interesting talk. So, the toxicology one is intriguing, right? Where that's the pathway we always go through, rats and all that. But, you know, I mean, I'm reminded of chocolate, that dogs cannot take chocolate, but humans can. But for every drug, we go through dogs, right? But how, so, I mean, is there a more rational pathway to not kill compounds too early just because that's the conventional way we've done things?
So everybody knows about difference between rodents and humans, and that's why the screening of this antigen and adjuvants needs to have at least a theoretical experiment bridge between the human and rat or mouse or bigger animals. No one knows probably the right answer for that yet, but now in vitro and in vivo and bridging organoid or similar experiment model is now being developed by many people. So we can try to make it, but most of the organoid lacks the immune cells. So that's really tough. And if you can make a experimental lymph node, I think we can give again one more Nobel Prize to those people.
Thanks.